A group of researchers out of Switzerland just made some really cool discoveries about the mitochondria. We're going to talk all about that and so much more on today's episode of Short Stories of Bacteria. So one of the best known facts, probably the best known fact in all of biology, is that the mitochondria is the powerhouse of the cell. Whenever someone says, "So what do you know about science?" then I feel like the natural reaction for many of us is, the mitochondria is the powerhouse of the cell. Yes, that's all very, very cool. But the mitochondria is so much cooler than just the powerhouse of the cell. And that already is kind of cool. But it's so much cooler than that. And the reason why is it has a lot to do with bacteria and evolution and endosymbiosis. We're going to jump into all of that in just 1 second. But first, welcome back, every single one of you guys, to another episode of Short Stories of Bacteria. I, as always, am your host, Dr. K, and I'm here to guide you through yet another story chronicling the lives of the tiny and the infamous, and of course, the bacteria and all such related microorganisms. If this is your first time, don't forget to hit that like, share, subscribe button if you're listening to this podcast on YouTube, or if you're listening to this podcast on some other medium such as Spotify or Apple Podcasts. Don't forget to hit that follow button to help support the podcast and help support the channel. But enough of that. Let us jump into the science. So, as I mentioned, the mitochondria is indeed the powerhouse of the cell. That's something that I think all of us know. Probably the mitochondria does tons of different things in the cell. The most important thing that we talk about is this. Honestly, this is probably the second most well-known fact in all of biology, and that is that ATP is the currency of the cell. So the ATP is a molecule, right? And you basically use individual molecules of ATP as a way of doing stuff in the cell. You can think about it as energy currency. You can think about like mana, however you want to think about it. If you want to do a small thing that only takes a little bit of energy, maybe you'll use 5 molecules of ATP. Or let's say you have a really, really big task that you need to do and you need to use thousands and thousands of ATP. Right? So ATP is the energy currency of the cell. It's the mana of the cell. And the mitochondria is the source of that, right? So if we don't have mitochondria, we don't have a way of, or at least you don't have a really effective and efficient way at producing tons of ATP, right? And so the mitochondria is extremely important. Now, there are many of you, many of the listeners of the channel /podcast know this, but if you don't, the current going theory, right, is that our initial mitochondria, came from a bacteria, right? And it's not just mitochondria. It's also something called the chloroplast, which is found in plant cells. But as far as we're concerned, the human beings, mitochondria is what we have. And it's theorized that these came from bacteria. And so how this is theorized to have happened is you have some kind of eukaryotic cell, or almost a eukaryotic cell at this point. Maybe it's developed some organelles, but not other organelles, right? And what it did is it trundled upon, or it happened upon, a bacteria. And we'll talk about it later, the type of bacteria, but for now we call it alpha proteobacter. It's described as alpha proteobacter, right? And what it did is it swallowed up this alpha proteobacter bacteria, and then this bacteria figured out a way to have some kind of symbiosis with the newly formed or emerging eukaryotic cell, right? One of the things that it did is it is involved in ATP synthesis. It's making more ATP. And so this newly formed eukaryotic cell is like, oh, well, this is very useful. I now have a way of almost like extracting or pulling ATP from this new bacteria that's kind of hanging out inside of my cytoplasm, right? And so and of course the mitochondria, now it's enclosed in the cell and it's being protected from any kind of predators or anything like this, and it's obviously not getting killed by the newly formed eukaryotic cell. And so they formed what's known as an endosymbiotic relationship, right? And it's from this endosymbiotic relationship that our current relationship with the mitochondria emerged. So right now, we are all eukaryotic. We're composed of eukaryotic cells, and every single one of our eukaryotic cells has mitochondria in them. Different cells will have different amounts of mitochondria. There's different sizes or shapes even of mitochondria as well, and the mitochondria still have many characteristics that it has in common with the bacteria, which we'll talk about in a second. But it's important to note that all of our evolution, the evolution of our entire human species, right, is contingent on this moment when an emerging eukaryotic cell absorbed a mitochondria or a bacteria that became the mitochondria, and now we use it as the source of energy. Now, all this may sound a little far-fetched. Maybe it's like, well, eukaryotic cells always had mitochondria, or they just kind of emerged with mitochondria in their cells. So what is it that makes us actually think that this was a bacteria, that these mitochondria were originally bacteria? And there's a couple different through lines here. The first one, obviously, is the shape. If you look at the shape of mitochondria, it looks very, very similar to an actual bacteria. It makes a little rod-shaped thing, and bacteria have a little rod-shaped thing. And this isn't a great argument on its own because it's much smaller than the actual eukaryotic cell. Bacteria are much smaller than eukaryotic cells. Now, this isn't the best argument. I mentioned a little bit earlier that they can come in different sizes and different shapes. So it's not a great example, but it does kind of. It's one thing that would make us pause, right? It's like, oh, okay, so maybe I could squint and be like, oh, maybe that is a bacteria or maybe that's a mitochondria. But there are also some other really good reasons why we think that they are emerging from bacteria. Another reason is that the lipid composition of the membrane in mitochondria is actually nothing like the lipid composition of our own cell membrane, right? You wouldn't expect that to be the case necessarily, right? And there are some similarities between the lipids in the membrane of the mitochondria and the lipids that are in the membranes of different kinds of bacteria. So that's another indication. Also, there are similarities in their DNA as well. So this is a real quick side quest, but you may not know this, but mitochondria has its own DNA that is distinct from our own normal genome, right? It's distinct from our own genome. The mitochondria has its own genome, and this genome is actually very similar to genomes that we see in bacteria. The mitochondrial genome, or the mitochondrial DNA, it forms a circle as opposed to chromosomes like we see in like big old chromosomes with chromatin like we see in our normal genome. Mitochondrial DNA also lacks something called introns, which is a very common trait in bacteria, and the overall what I'll call mitochondrial genome is very, very small. It's even smaller than many bacterial genomes, which is kind of wild. But all of this is pointing to the fact that the mitochondrial genome, it certainly doesn't look anything like the normal eukaryotic genome that we have in our nuclei. And in fact, it looks a lot more similar to a bacterial genome than it does to that normal nucleus genome that we have. Okay. Now, the last thing that I think is absolutely wild is that there are similarities between how the mitochondria and how bacteria interact with the immune system, right? So our immune system has or senses the presence of bacteria by things called PAMPs. So all these PAMPs they're these molecules that are associated with pathogens, right? And the immune system recognizes those and uses that as a trigger for itself. So it recognizes a PAMP and then says, oh my gosh, there's a pathogen present. It's time for the immune system to go and flock to this spot and then try and kill the pathogen. This is all very cool, and it's very important that we have this so that way we can respond just like that to any incoming pathogens, right? Now, what's very interesting is that the mitochondria has something very, very similar to PAMPs. They have these things called DAMPs, right? And all of these are doing, they do essentially the same thing. If these DAMPs are exposed to the immune system, then the immune system will respond as if there was a pathogen in that location. Okay, I'm going to say that again. If these DAMPs on the mitochondria are exposed to the immune system will respond as if that mitochondria is a pathogen. That's so, so cool. It doesn't matter. We've been hanging out with mitochondria for a very, very long time, but our immune system still recognizes these components as being similar to a pathogen. Okay? This is actually why, this is a real quick side quest, this is actually why when a cell dies, it undergoes something called apoptosis, where it has to, in a controlled manner, die. This is one reason why it's really important that the cell dies in a controlled manner, right? Because it has to safely sequester these mitochondria so as it's not triggering the immune system. So that's very, very cool. So that's our second big point. They seem to be a whole lot like bacteria. Now, all this is very, very cool. It supports this hypothesis that we got our mitochondria from bacteria through some endosymbiotic relationship. But a really, really good way, this only goes like part of the way, a really, really good way of demonstrating that the mitochondria came from bacteria is if we could replicate this symbiosis event again. If we can replicate this symbiosis event, this would be really, really good evidence that our mitochondria came from a bacteria, because it would show that this can actually happen in the lab. Right. So let's talk a little bit about where the initial bacteria came from. So our initial bacteria that was the basis for a mitochondria is currently theorized. I mentioned earlier something called alpha proteobacter. It's suggested that it's related to a genus known as Rickettsialis, right? So this Rickettsialis bacteria then was schlooped into an archaea, right? And the archaea is suggested to be, and this, hang on a second, I'm reading this one. This is an absolutely wild type of archaea. It's called the Asgard superphylum. I clearly don't know much about archaea, guys, but regardless, Asgard superphylum, that sounds pretty wild. But the main point I'm trying to make here is that these styles of bacteria, right, and this style of archaea, they're long gone. Right, they're way, way back in evolutionary time. So we obviously can't take those guys and then bring them up and like kind of put them together and see if it works. So what we have to do is we have to have some other kind of model organism or series of model organisms that we can try and put together as a way of replicating this symbiotic event. Now, at the time of this paper, and it's about time that we bring in this paper here because this is so cool. This comes out of Switzerland, the Institute of Microbiology, in Zurich, Switzerland. Good job. This came out in October 2024. Now, what's brought up here is that this has been something that's tried before. But as it turns out, modern bacteria don't play particularly nicely with their hosts. Oftentimes what will happen is they'll try and take the bacteria and then they'll put it into the host and they'll be like, all right, guys, go do endosymbiosis. It'll be great. But as you might suspect, these modern bacteria, essentially they just divide really, really quickly and then they murder their host, right? So you have to find very, very specific types of organisms that will play nice together in order to do something like this. And the one thing the researchers figured out that was very novel is they identified two organisms. There was a fungus known as Rhizopus microsporus. So this is a plant pathogen. And they also found a bacteria called, here we go, guys, Mycetohabitans rhizoxinica. Mycetohabitans rhizoxinica. What an incredible name that is. Anyway, so these guys actually have a somewhat symbiotic relationship already because the bacteria is able to protect the fungus from predation, right? And obviously the fungus is able to offer a location for the bacteria to be safe, right? So what they did is they were like, okay, let's see if we can take these guys and put them together. Now, the way they did this is actually really, really cool. So they essentially have to do surgery on the fungus, right? Funguses have a thick cell wall, right, made of chitin, right? And so what they had to do is the researchers had to take a needle and then stab it into the side of this fungus, and then then there's a whole series of pressure from the interior of the cell that's pushing outwards. And so what they had to do is they had to use an actual bicycle pump, and then eventually later on they had to use an air compressor as well to maintain pressure sufficient to the point where they could push the bacteria into this fungus, right? So I can only imagine what that grant looked like. Well, we're going to take this bicycle pump and then we're going to stab a needle into into a fungus, and then we're going to use this bicycle pump to inject bacteria. Very, very cool stuff. And of course, this is traumatizing for the poor fungus, but eventually what they figured out was that after the fungus was able to recover, the bacteria and the fungus were able to survive just fine, right? They were able to survive, they were able to thrive, they were able to go around and like kind of just live as they were. Now, there was a drawback in terms of how well they survived, right? So after a few generations, if you took this symbiotic fungus and bacteria and you pitted them against just a normal fungus, the normal fungus is able to survive better. But if you artificially selected for the fungus that had the bacteria in them and kept growing them, eventually they were just as competitive and just as effective at surviving as the populations that didn't have the symbiote. So what this is indicating a whole bunch of different things. But one of the most important things is that if you take a bacteria and if you stab it into this fungus, and if there's maybe a population bottleneck or some kind of environment where specifically that bacteria or that fungus and bacteria were required to survive, right? Then you can end up with a population that's extremely good at surviving and can then replicate essentially what we initially think the initial eukaryotic cells were looking like. So this is a really, really cool example of how this initial endosymbiosis could have happened. It's really, really neat. Absolutely wonderful job from this squad out in Switzerland. But let's quick recap, and then we can get out of here. So number one, mitochondria and chloroplasts are theorized to have come from bacteria as eukaryotic cells swallowed up these bacteria, and then they became these targeted organelles. Two, these organelles, specifically mitochondria is the one that we talked about, still have a lot of things that are in common with bacteria. We talked about similarities in the genome, similarities in lipid composition. We talked about the impact with which the mitochondria touches the immune system. Our third, point number three, a way of demonstrating this how do I phrase this? A proof of concept for this endosymbiosis is if we could do that endosymbiosis again, right? So a lot of folks are still trying to replicate this endosymbiosis by taking bacteria and pushing it into some kind of eukaryotic organism. And finally, our fourth point, a group out of Switzerland did surgery on a fungus with a bicycle pump, an air compressor, and a big old needle. And then with that and the bacteria, they were able to replicate an endosymbiotic event, eventually creating an organism that had the ability to survive and thrive with a bacterial symbiote. Really, really cool example of evolution. Really good example of how we're drawing the net around maybe how our initial evolution occurred, how the initial cells showed up and what they looked like and how they were formed. So yes, but anyway, guys, that is all I have for you today. Thank you so much, each and every single one of you guys, for tuning in. And I will see you guys again next week on another episode of Short Stories of Bacteria.